Today we will discuss about a new topic, that is gram staining of bacteria, principle and procedure. This topic is included under the paper biofertilizer SEC paper for second semester NEP students. So before going to detail, let me give a brief introduction on gram staining. So this gram staining method was first discovered by a Danish scientist, bacteriologist Hans Christian Gram in 1884. Okay, so this technique is used to identify, to classify and to differentiate bacteria. So all of you know that in an ecosystem there are different types of bacteria present. Okay, so to classify this different type of bacteria, we have to first go for a basic technique that is called gram staining technique. So this gram staining technique, it is most common, important, and it is mostly used for differential staining techniques in microbiology. Okay, so this technique, it helps in identification, classification and differentiation of bacteria. So next, principle of gram staining. Already I mentioned that in the ecosystem we have different types of bacteria. So for basic classification, if we go for a gram staining method, we will have two types of bacteria. One is your gram positive bacteria and another one is your gram negative bacteria. Okay, so first let me discuss about the gram positive bacteria. In gram positive bacteria, when the bacteria is stained with primary stain, that is crystal violet, and when it is fixed with the mordant, that is the iodine, some of the bacteria are able to retain the primary stain. Okay, after decolorization, means after addition of alcohol also, the primary stain is not released from the body of the bacteria. Okay, so that is called your gram positive bacteria. So this gram positive bacteria, that is this one type of bacteria, it will retain the color of the crystal violet, that is purple color or blue color. It retain that color after addition of alcohol also, means after decolorization also, means if we want to decolorize the stain, then also the color of the crystal violet, it will not go out from the cell of the bacteria. Okay, why it happens? Because the cell wall of gram positive bacteria have a thick layer of protein sugar complex, that is called peptidoglycan, and another content, that is lipid, it is very less in the cell wall of gram positive bacteria. So what happens when we add the crystal violet and the mordant, that is your iodine, when we add these two compound, these two will bind to form a complex which is called crystal violet and iodine complex. Okay, so this complex, it will remain in the cell wall of the bacteria. Okay, so after addition of the alcohol, the cell wall, it will dehydrate, it will shrink, and as a result, the pore size of the cell which are present in the cell, the pore size of the cell wall, it will become very small. Okay, so as a result of shrinkage, as a result of addition of the alcohol, the cell wall, it will shrink, and as a result the pores present in the cell wall, they will become smaller in size. Okay, then because of this small pore size, it prevents the crystal violet and iodine complex to come out from the cell wall. Okay, so as a result, this complex remain present in the cell wall, and when we observe under the microscope, finally we will see the purple color of the crystal violet. Okay, so this is the main principle, this is the main chemistry behind the purple color of the gram-positive bacteria after gram staining. Okay, and next, what happens to gram-negative bacteria? In case of gram-negative bacteria, cell wall, it has peptidoglycan but very less quantity. Okay, so the cell wall, it has very less amount of peptidoglycan. As a result, what happens when this crystal violet and iodine is added, it forms a crystal violet and iodine complex, but due to the thin layer of peptidoglycan, already I have mentioned that peptidoglycan is very less thin layer. Okay, and the thick outer layer, that is your lipid. Okay, so as a result, the crystal violet and your iodine complex, it gets washed off when we add alcohol. Okay, if we add alcohol, then the cell or the cell wall, it will shrink. Okay, but as the peptidoglycan is very thin and lipid amount is more, that is why the pore size will not become smaller. Okay, buji paisa, have you understood? So as a result, all the cell wall, I mean the pore size, will remain same because the peptidoglycan is very less. The shrinkage will be less. So as a result, when we washed off the stain, the peptidoglycan and the iodine, crystal violet and iodine complex, the complex, it will come out from the cell wall of the bacteria. Okay. So that is why when we again add the counter stain, that is safranin, then that safranin will present in the cell wall of the body. So the process, what is the method of gram staining, I will come to it later on, but let me first discuss about the principle. So because of the presence of peptidoglycan, high amount of peptidoglycan, so the crystal violet and iodine complex, it remains in the cell wall after de-staining also. But in case of gram-negative bacteria, because of the less amount of peptidoglycan, the crystal violet and iodine complex, it will come out from the body of the bacteria after addition of alcohol or decolorization. Okay, then if we add safranin, the gram-negative bacteria, it will again take the safranin color. So that is why when we observe under the microscope, we can see the gram-negative bacteria, they are red or pinkish in color. So next, coming to the reagents used in gram staining method. First, already I have mentioned that crystal violet, it is the primary stain. Then we add iodine as mordant. Then a decolorizer made of acetone or alcohol, that is 95% alcohol. Then the last one is your safranin, that is your counter stain. Then coming to the procedure of gram staining, how we can do the gram staining method to identify a bacteria. Okay, so first we have to take a clean grease-free slide. Then we have to take a smear of the bacterial suspension. Okay, a bacterial suspension we have to prepare on the clear slide, which is called your smearing. Okay, means on that slide we have to take another slide at 45 degree angle, and we have to smear it. Then it will form a clear thin layer on the slide, that is the bacterial film will be produced. Okay, then this film or the suspension is air dried and heat fixed. Heat fixed, that heat should be very low flame. Then crystal violet is poured and it is kept for about 30 seconds to 1 minute. After keeping it for 1 minute, we will rinse it with water Then the suspension is then flooded with grams iodine, which is your modern, for 1 minute, and again we will wash with water. Then the whole slide, it is washed with 95% alcohol or acetone for about 10 to 20 seconds. Okay, again we will rinse it with water. So this alcohol, it will dehydrate the cell, it will decolorize the cell. Then after that we will again add the counter stain, that is safranin, and we will keep it for 1 minute, and again we will wash it with water. Then we will dry the slide, then we will blot it and we will observe it under the microscope. So this is the whole process of gram staining. So next, coming to the interpretation. After gram staining method, what we have seen that some bacteria, they are purple in or bluish in color. In the first figure you can see that is gram positive bacteria, and in the second figure you can see that the bacteria, it is reddish in color. These are the gram negative bacteria. Okay, so why it happens? Because the gram positive bacteria, they have peptidoglycan, high amount of peptidoglycan, thick layer of peptidoglycan in their cell wall, which retains the crystal violet and iodine complex. After addition of alcohol also, it doesn't leave the crystal violet color. But in case of gram negative bacteria, as the peptidoglycan layer is very thin, that is why after addition of alcohol or acetone, it will lose the color of the crystal violet, that is the complex, it is released from the cell wall of the gram negative bacteria because the cell wall is very thin. Okay, so then again we will add safranin, that is why the cell, gram negative bacteria, the cell, it will take the safranin color. That is why you can see the color of the safranin in case of gram negative bacteria. So this is all about your gram staining method, the principle and the interpretation. Why we go for gram staining method, why it is important, and how we can see, how we can observe the bacteria under microscope, how we can classify the bacteria. Hope you have understood. Thank you.